Chapter 14. Hormones and Lipid Transport. Lipid transport is directed by hormonal signals that determine when fats are stored, when they are released, and how they are distributed between tissues. After lipids are absorbed and packaged into chylomicrons, they enter circulation in a form that requires further regulation before they can be used. Hormones act on both the transport particles and the receiving tissues, controlling the rate at which fatty acids are extracted and the destinations to which they are delivered. This regulation links digestion to systemic energy use and structural maintenance. Insulin plays a central role in directing lipid flow after a meal. When nutrient availability rises, insulin signals tissues to take up incoming substrates and promotes the storage of fatty acids. It increases the activity of enzymes that allow tissues to extract lipids from circulating particles and incorporate them into storage or structural pools. This process reduces circulating lipid levels and shifts the system towards storage. The strength and duration of this signal determine how much of the incoming lipid load is retained within tissues. Counterregulatory signals shift the system in the opposite direction. When energy demand increases or when nutrient availability declines, hormonal signals promote the release of stored lipids and reduce the uptake of new ones. This change alters how transport particles are processed, allowing fatty acids to remain available for use in energy production. The balance between these signals determines whether lipids are directed toward storage or toward immediate utilization. The interaction between hormones and lipid transport particles controls how quickly lipids are cleared from circulation. Chylomicrons deliver fatty acids to tissues through enzyme-mediated processes that are influenced by hormonal state. When signals favor uptake, these particles are processed rapidly and their contents are transferred into cells. When signals reduce uptake, the clearance of these particles slows and lipids remain in circulation longer. This change affects both the timing and distribution of lipid delivery. Hormonal control also influences how different tissues receive lipids. Some tissues increase uptake under specific conditions, while others reduce it, creating a distribution pattern that reflects current physiological needs. This selective allocation ensures that lipids are delivered to areas where they can be used for energy, membrane structure, or signaling functions. When hormonal signaling is aligned with tissue demand, this distribution remains coordinated. When it is not, lipids may be directed toward storage even when immediate use is required, or remain in circulation without being effectively utilized. The coordination between digestion and hormonal regulation determines how efficiently lipids move from intake to use. Digestion prepares and delivers lipid substrates into circulation, while hormones direct their fate within the body. When these processes are aligned, lipid transport supports stable energy availability and structural maintenance. When they are not, the movement of lipids becomes less predictable and their distribution no longer reflects the needs of the system. Hormones therefore act as the control layer that governs lipid transport beyond the intestine. They translate nutrient availability into directed movement and allocation, shaping how lipids are stored, released, and used. The effectiveness of this system depends on both the integrity of digestive processes and the accuracy of the signals that regulate distribution. Cholesterol and hormone synthesis. Cholesterol functions as a structural precursor for a range of signaling molecules that control growth, metabolism, and stress responses. Its role begins at the level of membrane architecture, where it contributes to stability and fluidity, but extends into the synthesis of steroid hormones that regulate systemic behavior. This dual function links lipid transport and cellular structure to endocrine output. The availability of cholesterol within cells determines whether hormone synthesis can proceed at a rate that matches physiological demand. Cells acquire cholesterol through both internal production and external delivery. Lipoprotein particles circulating in the bloodstream carry cholesterol to tissues, where it is taken up and incorporated into cellular structures or directed towards synthesis pathways. The balance between uptake and internal synthesis determines the intracellular pool available for hormone production. When delivery is consistent and aligned with demand, cells maintain a stable supply that supports both membrane integrity and signaling functions. Hormone synthesis begins when cholesterol is transported into specific regions within the cell where enzymatic conversion can occur. This movement represents a controlled step that determines how much substrate enters the synthesis pathway. Once positioned, cholesterol is converted into intermediate molecules that are further processed into active hormones. Each step depends on enzyme availability and cellular conditions, but the entire sequence is limited by the initial supply of cholesterol. The production of steroid hormones depends on the rate at which cholesterol can be mobilized and converted. When supply is sufficient and transport within the cell is efficient, hormone synthesis can respond to changes in physiological demand. When cholesterol availability is reduced or when its movement into the synthesis pathway is constrained, production declines. This limitation affects the output of hormones that regulate metabolism, stress response, and reproductive function. Cholesterol transport within the body also influences where hormone synthesis occurs. Different tissues specialize in producing specific hormones, and their ability to do so depends on the delivery of cholesterol through circulating lipoproteins. When lipid transport systems are functioning consistently, these tissues receive a steady supply of substrate. When transport is disrupted, cholesterol delivery becomes uneven, and hormone production can vary between tissues. The connection between digestion, lipid transport, and hormone synthesis becomes apparent when considering how cholesterol enters the system. Dietary fats influence the formation of lipoproteins that carry cholesterol through the bloodstream, and the efficiency of fat digestion affects how these particles are assembled. When lipid processing is aligned, cholesterol is packaged and delivered in a manner that supports stable distribution. When it is not, the availability of cholesterol for cellular use becomes less predictable. Cholesterol, therefore, serves as a central substrate linking lipid transport to endocrine function. Its movement from digestion to circulation and into cells determines whether hormone synthesis can proceed effectively. When delivery, uptake, and intracellular transport are aligned, cholesterol supports both structural and signaling roles. When any part of the sequence is disrupted, hormone production reflects that limitation, and the effects extend across multiple physiological systems. Lipid-based signaling systems. Lipid-based signaling emerges from the same structural framework that governs membrane composition and lipid transport. Signals are not generated in isolation. They arise from the physical state of membranes, the availability of specific lipid substrates, and the enzymes that convert those substrates into active signaling molecules. This creates a system where structure determines signaling capacity. When lipid composition is stable and substrates are available in the correct proportions, signaling proceeds with precision. When it is not, the signals themselves become inconsistent. Cell membranes act as the platform where many of these signals originate. The arrangement of lipids within the membrane determines how receptors function, how enzymes are positioned, and how quickly signaling molecules can be generated and released. Membrane fluidity and organization influence the interaction between signaling proteins and their lipid environment. When membrane composition supports these interactions, signals are generated and transmitted efficiently. When it does not, receptor activity and downstream signaling pathways become less reliable. Many signaling molecules are derived directly from membrane lipids. Enzymatic activity releases specific fatty acid components that are then converted into active mediators. These molecules act locally, altering cellular behavior in response to changes in the environment. The availability of these substrates depends on both dietary input and the efficiency of lipid digestion and transport. When the supply of lipid substrates is consistent, signaling output remains stable. When it fluctuates, the production of signaling molecules varies accordingly. The timing of lipid-based signaling reflects changes in substrate availability and cellular demand. Signals can be generated rapidly in response to stimuli, but their magnitude depends on the pool of available lipid components. When these pools are maintained through consistent digestion and transport, signaling responses remain proportional to the stimulus. When substrate availability is limited or uneven, signaling may be reduced or delayed, affecting how cells respond to changes in their environment. Lipid transport systems also influence signaling by determining how substrates are distributed across tissues. Lipoproteins deliver fatty acids and cholesterol to different regions of the body, affecting the composition of local membranes and the availability of signaling precursors. This distribution creates variation in signaling capacity between tissues, reflecting both their function and their access to lipid substrates. When transport is aligned, these differences support specialized functions. When it is not, signaling capacity may be reduced in some areas while remaining intact in others. The interaction between lipid digestion, transport, and signaling creates a continuous chain that links dietary input to cellular response. Digestion determines which substrates are available, transport distributes them, and membrane organization converts them into signals. Disruption at any stage alters the entire system, changing both the availability of signaling molecules and the conditions under which they are generated. Lipid-based signaling systems therefore depend on structural integrity and substrate availability. They are not independent pathways, but extensions of membrane architecture and lipid metabolism. When these underlying systems are aligned, signaling remains consistent and responsive. When they are not, the generation and transmission of signals reflect the same variability, influencing how cells and tissues coordinate their activity. Transport is regulation. Transport is not a passive step that follows digestion. It functions as a control layer that determines how quickly nutrients appear in circulation, which tissues receive them, and in what form they arrive. The structure of transport particles, the pathways they move through, and the signals that govern their clearance define the rate and pattern of nutrient delivery. This means regulation is embedded in movement itself. Changing how a nutrient is transported changes how it is used. Lipid transport illustrates this directly. After absorption, fatty acids are repackaged into chylomicrons that enter the lymphatic system before reaching the bloodstream. This routing delays their appearance in circulation and spreads delivery over time. The size and composition of these particles determine how they interact with tissues and enzymes that release fatty acids. Larger particles persist longer and deliver lipids gradually, while smaller or rapidly cleared particles shift delivery towards specific tissues more quickly. The transport structure sets the tempo of availability. Enzymes that act on transport particles add another layer of control. As chylomicrons circulate, their contents are released in response to tissue demand and hormonal signals. This release is not uniform across the body. Some tissues increase extraction under certain conditions, while others reduce it, creating a distribution pattern that reflects current needs. The same amount of lipid can be directed toward storage or toward immediate use depending on how these transport interactions are regulated. Movement through the system becomes a mechanism for allocation. Water-soluble nutrients follow a faster route through the bloodstream, but their transport is still regulated by carrier systems and concentration gradients. Rapid entry into circulation increases availability, but uptake into tissues depends on transport proteins and signaling states that control entry into cells. When transport into tissues is limited, these nutrients remain in circulation longer or are redirected towards storage pathways. The speed of transport therefore influences both availability and fate. The timing of transport also determines how nutrients interact with one another. Lipids delivered through chylomicrons arrive over an extended period, while glucose and amino acids appear more rapidly. This difference creates a sequence of availability that affects how the body processes each substrate. When these timelines are aligned, energy use and storage remain coordinated. When they are not, nutrients compete or are redirected, altering the overall pattern of metabolism. Transport pathways also influence where regulation occurs. Lipids passing through the lymphatic system bypass immediate hepatic processing, allowing peripheral tissues to receive them first. Water-soluble nutrients pass directly to the liver, where their distribution is modified before reaching the rest of the body. These route differences create distinct regulatory points that shape how nutrients are handled after absorption. Transport therefore acts as a regulatory system by controlling timing, distribution, and interaction between nutrients. It determines whether substrates are delivered in a steady flow or in rapid pulses, whether they are directed towards specific tissues or dispersed broadly, and whether they are used immediately or stored. The structure and behavior of transport systems define how digestion translates into physiological outcome, making movement itself a primary mechanism of control.